Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
Welcome to another past HSC exam question videos. In this video we're going to cover this exam question, which comes from the Biotechnology chapter. In a second I'll read the actual question. Once I read the question, you have about 5 seconds to pause the video and then to attempt the question, and then when you're ready press play and I'll go through the actual answer itself. The question is: traditionally, banana plants in Australia have been propagated asexually by cutting out a planting sucker from the adult plant. And this here is the planting sucker, this one here. There is a growing trend to produce disease-free plants in laboratories through a process of cloning from disease-free tissues from existing plants. Assess the potential impact of this cloning process on the genetic diversity of banana plants in Australia. And that's worth 4 marks. So when you're ready, pause the video. Welcome back. Alright, so you should analyze, what is this traditional technique that we used? Well, it's about asexual reproduction. And asexual reproduction means that the parent is identical to the offspring. That's asexual reproduction. Now it's saying this new trend is that we use this new technique which produces new plants through a process of cloning. Cloning is also asexual. So it's using cloning, which is asexual, from diseased tissue from existing plants. In this case, it's also parents are identical to offspring. So what we have to do is we have to assess the impact of this technology on the genetic diversity of the banana plants in Australia. And I made the claim that there wouldn't be much of a difference because we used to have asexual reproduction, which means that the parents are identical to the offspring. And the new technology is the same thing. The parents will be identical to the offspring, especially as it says that we use existing plants. So we will use the ones, the banana trees which are already in Australia and use technology on that, which means really there won't be much of a difference in terms of diversity. So that's what I discussed in my video here. That's what I'll show you. First I wrote, there will be very little impact on the genetic diversity of the banana plants. That's my statement. Traditionally, the plants were clones of the parents as they were reproduced asexually through the plant suckers of a parent tree. Right, so that's more or less cloning. Then what I wrote is new process would involve cloning disease-free plants from existing plants. This means that there's virtually no genetic diversity change, as both techniques rely on asexual reproduction of existing banana plants. So they both use the same, more or less the same original tree, and they both have the cloning technique. One is using it in the laboratory, the other one is using the shoots, which is still a form of cloning. And then, so that's more or less it. It's 4 marks, but if you write those statements, I guess this is more of a comprehension kind of question. So seeing that you should realize that there's not much of a difference. So get a mark for the correct statement here. You get a mark for mentioning that, this is actually a cloning, that the first method was also cloning. The second method is cloning. And that because they're both cloning, there won't be much of a difference. So we've explained how those two techniques are actually more or less the same. And where that question come from? It comes from this dot point. Discuss the potential impact of the use of reproductive technologies on the genetic diversity of species, using a named example of a plant and animal that have been genetically altered. So it's the first part, discuss the potential impact. In this case, you should realize that they're both asexual reproduction, which means they're both cloning. So there would be not much of an impact if we use that technology instead. I hope that was useful.
Thank you for watching.